Welcome, everyone. Thank you for tuning in today. My name is Simon Dia. I am project coordinator of the project ProxIMed, and today I am happy to uh, invite you to this webinar about nutritional aspects of alternative proteins. Thank you for tuning in. I will share a disclaimer shortly. Just have to enter my presentation. So, um, first of all, um, for everyone to know, this meeting will be recorded. I kindly ask you to agree to the disclaimer, as this is a European project. We are recording this meeting and to share our education and research according to open research data principles of the European Union. Therefore, I kindly ask you to agree. Oh, 1 second, and I will open the questionnaire. Oh, 1 second. All right, so I kindly ask for your consent. And it also will be in the chat for your convenience. And if you have any questions, you can come forward. All right. And I shortly want to introduce you to the agenda of today's webinar, Nutritional Aspects of Alternative Proteins. There will be 3 presentations covering neutral aspects of alternative proteins, and there will be a short break after the first presentation, and there will be a 5-minute question and answer round after each presentation. And therefore, I kindly ask you, if you have questions, you can type them in the chat if you don't feel comfortable opening your mic and your camera. Otherwise, you can just open up your mic and camera during the question and answer session. And my colleague, Nietzsche Erkan, will monitor the chat and forward contact details, or if you have specific questions or want to link up with the presenters to give you the contact details. And today we have the pleasure to hear from 3 outstanding experts in the field. One of them is Dr. Tullia Tedeschi from the University of Parma. She will explore the importance of amino acid score and addresses the digestibility challenges for alternative and plant-based proteins. Then we have Dr. Dimitris Fatouros from the Aristotle University of Thessaloniki in Greece. And he will present his insights into bioavailability and the absorption of bioactive substances in the gastrointestinal tract. And last but not least, we have Dr. Frederick Lia from the Malta College of Arts, Science and Technology in Malta. And he will highlight potential toxicological considerations in alternative proteins. And I will give a short introduction about our project and what we are actually doing. And I will start with a brief overview on that. So first of all, what is ProxIMed? We are focusing on implementing products with alternative proteins in the Mediterranean area. and we focus mostly on protein uh, products. And what is our mission? ProxIMed focuses on to revolutionize the protein consumption in the Mediterranean area through innovation and sustainability. And we focus on developing 20 alternative protein products. And we involve consumers, industry partners, and innovative technologies. And by reducing the environmental impact, improving the food security, and enhancing food choices. And we strive for more healthier and more sustainable future in the Mediterranean diet and for the Mediterranean population. And yeah, who we are. So we are 17 partners from 10 different countries. And in the following slide, you can see our partners. And we have partners from Turkey, from Germany, from Lebanon, from Greece, and from Tunisia, as well as from Egypt, Spain, Portugal, Malta, and Italy, of course, not to forget. And all this was made possible due to the incredible minds of our project leader, Professor Özlem Ismutlu, and the vice coordinator, Professor Mecit Öztop, as they achieved to get the project granted by the Prima Foundation, which is founded by the Horizon 2020 program and the European Union. So what do we do exactly? So we extract proteins from novel sources, agricultural byproducts, and traditional plants that are already part of the Mediterranean diet. We use novel processes as well as conventional processes, and we want to decrease the environmental impact using those technologies, create effective processes, and aim to improve functional properties while ensuring food safety and food quality. And to prevent from a theoretical scenario, um, we also implement market research to act according to consumers' needs in the Mediterranean area and implement those needs of the consumers into the product development. And as already mentioned, we are developing 2020 products and looking forward to entering the markets with those products in Turkey and in Tunisia. Here you can see a brief overview about the products that we are going to produce and the partners and countries involved in it. And as you can see, we have the agricultural byproducts, classical ingredients such as lentils or sesame cake, which are already present in the Mediterranean diet, but also novel resources such as insects, microalgae, or mallow leaves. And this brings me now to the next step of our webinar. I am pleased to introduce the next speaker, which will be Tullia Tedeschi, and I kindly open the stage for you, Tuya. Thank you very much.
Thank you. Sorry. Can you see?
Not yet.
Okay. Now it's on, I think.
I can't see it. I see a Zoom link now, or is it just me?
It says that I'm sharing the screen. I'm trying again.
Yes, there it is. Now I can see it. Thank you.
Okay.
So the stage is yours, Tullia.
Thank you very much. I'm sorry for this first problem. So, hello everybody. It's a pleasure for me here. My name is Tullia Tedeschi, and I'm associate professor in organic chemistry at the Department of Food and Drug, University of Parma, and I'm a partner in ProxIMed project, and as I'm the principal investigation for the unit of University of Parma. Today, as already Simon said, I'm talking you about some important feature relating the nutritional insights of plant-based and novel protein sources. So regarding the importance of amino acid composition, amino acid score, and how these address the digestibility, protein digestibility of these new important protein sources. No, not working.
Well, we know it worked previously.
Previously it worked. It's so
Maybe use the keyboard?
no, because I use it before.
Or Tuya, maybe you have to go back into the presenting mode in your PowerPoint. Maybe that's the case. So now we're in normal mode. Now we're in presenter mode. Yeah, and it's changing. Very good.
Okay, so what is the focus now? I mean, the focus is about proteins. We already know everybody what are proteins and why they are really important for our life. I mean, they are part of biomolecules that are really important for our body, and also food proteins. So the proteins that we can, achieve by our diet. And the same time, we know that there are many issues nowadays related regarding how is going on in our, in the world by the increasing of population at the same time, and also the fact that natural macro sources are decreasing. So we are, focusing on an expected increase in protein demand, which is really rising every year. And this is actually an aspect that has been to pose the attention on the switching new strategies and switching from animal proteins based to the vegetal one. Again, I think it's like a double matter. So from one side, food security. So the need of increase the protein demand, but at the same time is also a sustainability issue. As you know, most of the animal proteins that we acquire, they are coming from very intensive livestock. So this is actually a problem, like for energy consume and so on. So there is an increasing pressure on shifting from traditional protein sources to the alternative one. Actually, when we are using this adjective alternative, we are just to pay attention, because Alternative, it means that something that has not been used before. So, for example, if we are mentioning some plant vegetal sources like legumes or other kind of also just the list of proteins that Simon has already shown you, some of them are not really alternative, okay? So they are vegetal sources that I already introduced in different diets, but maybe some times ago we didn't think about them as a protein sources. Alternatives, okay, they are the new ones. So as we will see, mainly some of them are also called novel foods because they are really a new source of foods, new proteins, for example, coming from algae, insects, and so on. Actually, I think we can divide the plant-based alternative proteins in three groups. One group is the traditional alternative sources. So by mixing these two terms, traditional and alternative, we are thinking about some vegetal plants, some vegetal sources that they already introduced in our diet, but we didn't eat too much them by thinking about the protein intake. So the legumes, I think, is most of them sesame or other. At the same time, we have this kind of class of plant-based alternative proteins. They are coming from agro-industrial byproducts. This is quite interesting, and it's quite a new and focusing subject related to the sustainability. So if you think about the necessity, the means to recover and valorize protein fraction that can be extracted from the agro-industrial byproducts, so in a circular way of reutilization. Finally, there are some new sources. They are called novel foods. There is a strict European regulation about this. It was from the 2015, and I mean, some of them, they have never been used in Europe before. They are introduced now, and like insects or other kind of new proteins or new extracts from different sources, they, I mean, seems to be very promising in a way of shifting the perspective from an alternative protein view. What I mean, I try just here to stress, I think it's quite important that if we are considering a new product, I think that these 3 points are quite very important. So, for example, we have to be sure that when the consumer is going to buy this new product, the labeling is clear and not misleading. So it means that that should be written exactly what is inside of the food product. At the same time, of course, and this is actually the topic, the nutritional aspects are really important. So the product should have nutritional features which are comparable with the product they intend to replace at every level. So just think about the example, and we will see some of these, the example between the use of this alternative burger or vegetal burger made by soybean instead of the beef burger. So there should be, I think, a nutritional key strategies and say, yes, I can eat this because it's comparable from a nutritional point of view. So you will see, for example, the nutritional value, the essential amino acid intake is similar. At the same time, okay, the food should be safe. So the consumer must not incur in any risk by eating them. And this is very important. And also for some, aspects nowadays, some of these safety concerns, safety assessment are not really completed already for these new kind of products. Here, I just try just to show some theme, just to summarize and how we can a little bit classified, divided the different food proteins according to the source, so the animal, plant, and fungal. And you can see that, for example, insects. We talk a lot about insects nowadays. They are animal plant and animal protein source, but actually the interest of introducing insects in our diet is due to the fact that if you think about growing up insects, this is quite convenient. If you compare it by growing up breeding beef and so on. So this is actually one of the reasons, because we are thinking about, from a sustainability point of view, that the introduction of insect proteins in our diet would be an advantage. Okay. We also see there are different plant sources. They contain different kinds of proteins according to the different classes of protein and structure. And also the protein content is very different. So for example, cereals, different kind of cereals that are common use in our diet, they do not have such a very high protein content inside, and actually not too much, with not a very high nutritional value. Okay, but okay, legume family is the one that I just mentioned before, and also is quite common. There are different kind of legume, and they have, very quite high protein content. So at the beginning, I would say it appeared the best option to just to shift from animal to a plant one. Then, of course, we have this kind of different pseudo cereals or edible seeds. So some of these maybe were not so common in our countries to use, but nowadays they are becoming quite common. Quinoa or chia, since they have inside not only proteins with a very nice content, but also they show the presence of different secondary metabolites that they seem to have some kind of interesting, also beneficial properties. Finally, it appears that mostly in different vegetal fruits, for example, proteins seems to be stored mostly in seeds. So some of these, for example, apple seed, rapeseed, they have a quite high content of protein inside, and actually the identification of these plant sources, protein sources, is quite useful, because it can be just linked to the fact that these seeds most of the time can be a byproduct that are produced, for example, during agro-industrial production. For example, think about the production of oil. When you are producing the oil, some of the residues are the pomace, or also the residues of seeds. And in the case of the oil production, you just throw away. This is a waste, but it can become actually a new ingredient, new product, because you can extract protein from it and just try to valorize in a algae. Okay, microalgae or algae still were not until some years ago very common used in Western countries, mostly in Asia, but nowadays they are introducing in our diet. They have a very high protein content inside, and so they seems to be very interesting. And on the right side of the slide, I just put this table. This is just a list of the possible functionality that protein can have in a food composition. So it's important, that we can just select protein source, select for its nutritional aspects. At the same time, we are interested also to just keep the quality, the structural features of protein, because they are needed if you want to to set up, to produce new foods, and also some of these functionality are really important in order to get finally the desired product and also the right, let's say, interaction between proteins and the other ingredient inside. I just focusing here from one aspect that it's quite important. When you are extracting protein from different sources, you have vegetal sources in principle, but it happens or sometimes also in animal one. You have a mixture of different components, and you have to try the best solution to extract protein from these heterogeneous media. And the most common method that has been used since many years is the extraction in basic conditions, so in alkaline extraction, and by increasing pH. Here is reporting up to 12. I do not recommend to go up of this, but it's possible to stay lower. And also sometimes with the help of a little bit temperature modulation. So the alkaline medium has the ability to disrupt all the, let's say, aggregation and also the networking that can be inside in a vegetal extract, due mostly to the presence of fibers. So protein in a vegetal source is aggregate with fibers or polyphenols. So it's very important. It's possible to use like this this technique of extraction because it's really effective improving the yield of protein and to extract protein to the rest of the source. And when you have solubilized at this condition at a high pH, then by applying the isoelectric point, you are able to isolate protein as a powder. Isoelectric point of a protein is the pH value at which the protein shows the most insolubility. Okay, this operation can be a little bit tricky because if you are, extracting different proteins from the same matrices, and this actually happens quite often, they could have a different isoelectric point. So it's important to modulate it in order to promote the precipitation and then the isolation. For example, pH 4.5 is quite common as an isoelectric point for the storage proteins in legumes or in other vegetal sources. So this methodology is quite low cost, it's rapid, it's simple to carry out, can be also, I didn't say it, but it can be implemented. So I'm not speaking about laboratory scale, but can be implemented also at the industrial scale, large scale. So that's very useful because at the end of the day, we would like to obtain a powder, protein powder, in a very high amount, and then we can use it for different application and different kind of formulation. However, we have to say that the increase of pH and also sometimes also the use of the help of temperature increasing can cause degradation of amino acids inside of proteins, and this by losing some kind of functionality of also it can affect the quality of proteins. Here on the right you will find just a flow chart that just step by step they are a little bit explain how the steps that are made technically in order at the end to have your final product. Purification, if it's possible, if it's needed, is optimal to do, and also protein renaturation. We will talk about it just now. At the end, you can obtain this protein powder that it's commonly called concentrate or isolate. The denomination of concentrate or isolate depending on the protein content inside. Usually, concentrate is defined at up to 60% of protein inside. Isolate is up than 80%. So what we can say, what are the key factors that are important if you are thinking to replacing the animal proteins with plant-based alternative analogs? So we have to, we think we have to just to have some questions and to try to answer in order to do exactly the comparison between the two different sources. So first of all, we have just seen does the extraction method affect the protein quality? So it's important to select the right extraction method, which can, guarantee the quality of the proteins product, and then the possible and the future use in the future application. So we will see also the importance of, at a nutritional point of view, the amino acid profile of the protein that we are extracting, and see if there is different, the presence of different amino acids, and how this is different from, a reference point that could be, for example, I don't know, the egg proteins. Usually, in the past, egg protein was considered the gold protein because inside there is a distribution of amino acids that is really very well balanced and ensure the high nutritional value. Finally, it's important again to see, we see the first step, we got the ingredient, we characterize and we have a nutritional, composition. The ingredient is used for produce new foods. What's happening to the food when it is ingested by us? So the digestibility, protein digestibility, so the possibility of the different protein fragments to be absorbed, and the colleague after me will describe you very well this, what is happening during, after digestion and during the step of absorption. But of course, it means how is the bioavailability of these new proteins compared with the animal ones. And also this is quite important. And technically speaking, some models have been designed in order to give some indications about this. So when here is just to sum up what I have already told you. So the extreme pH, the high temperature, high pressure can affect the protein structure and give some modification. Also high pressure, just think about different processing technology that can be applied, for example, also to obtain a very dry component. So after the step of extraction, food industry, before using the ingredient, apply some kind of processes in order to get the very dry powder that can be used. Sometimes also these processes, if they are not really well controlled, they can well monitored, they can of course affect the structure of the protein. Which are, let's say, the key factor that we have to keep in mind during the extraction. So first of all, and I think that is the most common one, is the loss of the native structure. So when you apply high pH, what is happening is that the three-dimensional structure of protein can be lost. And this phenomenon is called denaturation. The denaturation can lead to, let's say, to final products. One is the simple unfolding. So the protein has a three-dimensional structure, and when it's treated by high pH, this three-dimensional structure is lost. And finally, you will find an unfold structure. Unfold proteins can be, for example, can lose some kind of chemical physical features, like, for example, solubility. Or we were mentioned before different functionalities. So the importance of using protein inside foods for their techno functionality. So if the three-dimensional structure is lost, the protein could also have lost some of the properties that are important when you are mixing proteins with other ingredients and you want to have a final food. Second, the high pH could also increase the tendency to proteins to aggregate one to the other one. So the aggregation process, and this aggregation process give rise to very, let's say, quite big molecules. And I think mainly the consequence is a loss you are losing the solubility. So this is actually a problem. So the loss of the native structure is not going to alter, let's say, the real chemical structure of a protein, but it alters the three-dimensional structure and then can affect different properties. So it's really important that when you apply this kind of process to obtain your ingredient, you keep an eye about the process and how the preserving the protein structure, because this can eventually affect then the quality of the protein and also the nutritional aspect. We know that amino acids are the main components of protein. 20 amino acids are incorporated of protein, and only 20 are in the genetic code of life. And they produce different combinations can produce different amino acids. It was studied in the very early 90s, so in the last century, how first studies how the intake of foods which are rich in proteins can alter, let's say, the different life cycle of different organisms and so on. Most of these studies, as you probably know, were made on animals. Okay, so this is quite just an example. I put it because I think it was quite impressive. There was this, scientists, and they studied by feeding dogs with different kind of foods containing different amino acids. He just realized that if he just took away tryptophan from the diet, this could give a negative nitrogen balance at the end for the dog by just analyzing the fecal, and so just analyzing and just give some hypothesis of the absorption, but at the same time, the same behavior was not observed if they just eliminate proline from the diet. So this is, from this experimental data, Aberdeen classified the amino acid into classes: the nutritional essential amino acid and non-essential. So the essential ones are amino acids that are not produced by our body. We need to get them from foods, so from our diet, and they are really important because they are needed for they are needed, they are absorbed by our body, and they are needed for any other steps of our cell and lives and so on. So you see, it's really important that the presence of these amino acids is kept in order to guarantee a very high nutritional value of the protein products that we are introducing in our diet, that we would like to introduce in the market. So this is the aspect. So to have an idea of the amino acid profile in order to give also an evaluation of the nutritional value. And if the amino acid profile is in some way altered by, for example, the introduction of different processes, this affect the quality of the entire product. So sometimes oxidation reaction of some amino acid side chains or other kind of modification can affect amino acid structure. So this can be observed, for example, in sulfurous amino acids like cysteine or methionine, which are essential amino acids, or sometimes also the degradation affected the stereochemistry. So we know that amino acids are all in the L form, and natural amino acid, by increasing temperature and pH, you can promote the racenization of amino acids, and the presence of the D amino acids can just let's say affect the absorption amino acids. So the D amino acids are not the natural ones, so they are not being recognized by our body. So this is actually eventually another kind of let's say alteration that can just lower the nutritional part and the quality of the protein. How we can evaluate the profile? So we can, estimate the amount of amino acids in a product. We can also estimate each amino acid inside, but then we need to compare this amount with a reference or other kind of let's say standard reference, in order to have an idea, an indication about the nutritional value of our product. And in this particular case, we would like, we need to compare the content of essential amino acids in protein, in alternative or vegetal protein sources, to the animal one, because you remember at the beginning we said the goal is the shifting between the two different protein sources. And for this reason, there are some protocols, some, let's say, standard calculation that can be made by using the amino acid score, also some other scores that are called PDCAS or DEAS. There are, they are quite different. So I'm just trying to explain a little bit about the difference between them, just simple, in order to, just to understand which kind of information this, score can give us and can be useful for, the implication that we say at the beginning. So the protein digestibility corrected amino acid score was introduced many years ago, and it's based on the estimation of the digestibility over the total digestive tract. It means that you are sampling, you are made fecal sampling, and then you are going to analyze and quantify amino acids inside this sample. Actually, and by the difference of the amino acids that you analyze, you can calculate the absorbed ones. Actually, it's quite, well recognized that this methodology generally overestimates the amount of amino acid absorbed. For this reason, more than 10 years ago, a new method was proposed, and is the digestible indispensable amino acid score. In this case, was proposed by FAO. I didn't mention before, but FAO periodically gives many reports, and updating these reports by reporting all this information about nutritional aspects of foods. And as you can imagine, this kind of reports have been updated recently with the use of, for example, vegetal foods and so on. For DIAS, we are going to consider the digestibility at the end of the small intestine, and it's quite well recognized that this measurement is more accurate than the one of PDCA-S. And but still, if you are using these two methodology, you have to think about that. You will just have an identification and quantification of amino acid profile in your product, and then you compare them with this pattern, amino acid reference pattern requirement given by FAO, and also the digestible amino acid amount. And you can have this data by, referring different, for example, databases where this digestible amino acid has been proved experimentally, and the in vivo tests usually are done on pigs or rats, for example. For example, I remember there is quite very well updated database made by INRA, the Institute of Agriculture and Food Research in France, and they reported the data reference for each amino acid for different proteins. There is for animal, there is for vegetal, and also nowadays, I think recently, they also updated this data for insects, for example. Some insects that have been already authorized by the European regulation, and they also calculated this data. So if you got this reference, then you compare with your protein, amino acid content in your product, and you can have an estimation. The difficulty in applying this two models is the fact that when you are considering a very complex mixture, so a food that is made by different protein components coming from different sources, so it can become quite a problem to estimate, to have an estimation of this data, unless you are going to do by yourself the experimental analysis. So you by yourself, you produce your new foods, and then you give the foods to be fed by the pig, and then from the pig you just the ileum extract the samples and analyze directly. In order to overcome this problem, there is a third possibility, and it's the considering the amino acid score. The amino acid score gives you an indication of the ratio of essential amino acid present in your food. So it's just give you the benchmark before. So it gives potentially what is the essential amino acid distribution in food, and so what is the distribution, the profile that is present in food and that can be absorbed in our body. And it is calculated based on the amino acid content that you can determine experimentally in food, and then compare it with the reference protein requirement that is made by FAO. And we will see a table later on. And usually these tables are just divided for each fragment of population. So considering the early birth child, young children, and also adult. And I think that they are working also nowadays to implement it also for the elderly people. This is actually quite a new issue that needs to be, of course, considered, because our population is getting older, and the life, actually, our expectation of life is increasing. What are the list of essential amino acids? Here are reported. Usually, for some cases, when do calculation, sulfur are mixed, are considered together, and also the aromatic one are considered together due to the different properties. Then you will see some of them are which are called aliphatic branched amino acids. So isoleucine, leucine, valine are the ones that are present in the protein powders that are used, for example, for physical body and some fitness activities. And also histidine, threonine, lysine is quite important. We will see that, for example, as I mentioned at the beginning, different protein sources coming from different organisms can have a different profile of amino acids. So, for example, you will find that some kind of vegetal sources are most rich in lysine and other one in cysteine. So, new trends is the idea to combine these two different protein sources in order to increase the composition, to better increase the composition of essential amino acid inside. Okay, because one product is lacking cysteine, the other protein is lacking lysine. By combining them, I can increase the nutritional value of the product. And this information, you can get this information by determining the amino acid score. So by determining the amino acid score, you can have an indication of the distribution of the amino acids inside of your product. And then this can be useful in order to understand if, for example, you are using this for the production of new products, to combine, to mix it with, to blend with other protein sources. And here are some calculations are reported in this slide. So there is a particular, you have to consider the single amino acid content, milligram per gram of protein, and also you can compare it to the specific reference that you will find, you see, for the different segment of population. And you can do, comparing with the FAO requirement and see what is the difference. So how is behaving your product. At the same time, for example, since we are speaking about shifting from animal to vegetal, in some works it appears the use of a reference protein. Reference protein, for tradition, nowadays is from animal. So, for example, I mentioned before, in different works actually, we use the egg as reference protein, no? Because egg is the gold food. So the best distribution of essential amino acids inside. So you can compare the distribution of essential amino acids in this, for example, new vegetal, new alternative protein extract, and compare to egg and see what is the improvement or what is the lack. Okay. Usually, most of the times, and this also give you, the indication of the the limit, because you got, I mean, the limiting amino acid value by calculation of the amino acid scores. And it means that, I mean, we know that by introducing this food in our body, this amino acid is not too much. So there is the possibility that cannot be available for our body for next steps. So why not combining the protein with another protein, which I know that is rich, for example, in cysteine. This is, for example, made by blending legumes with cereal, because sulfur amino acids are limiting amino acids in most of the legumes protein sources. On the other hand, they are rich in lysine, but on the other hand, for example, wheat or cereals are quite poor in lysine. So that could be a good combination. And we saw that on the market, some of this combination has been already used by a food processing approach. So, and I think it's quite very interesting because you can combine nutritional aspects also to processing features. So by combining these aspects also, you can also realize new ingredients and also just using the techno-functional of protein. Here I was just mention an example because I think it was quite useful. This was quite a recent work we published last year. We were interested in analyzing different new vegetal protein products that are on the market that are sliced cure meat. So, for example, we are from Italy, so you know that here in Italy, cure meat like prosciutto di Parma is a very well-known and traditional food. But anyway, at the same time, that we know that is a meat, pork meat, but is quite high in fat and also in salt. So that would be, I mean, that was the idea, no, to replace this kind of product with another kind of sliced meat product made by the combination of different vegetal proteins. In the picture here are reported. I mean, on the left, this is ham, cooked ham and bresaola, which is another typical Italian cure meat. And here we found on the market 3 different examples. They were produced by mixing soybean texturized proteins with gluten and other wheat proteins. We did, I mean, this work, we analyzed the protein profile, we analyzed the amino acid profile inside, and we estimate the essential amino acid content. And then we calculated the amino acid score. Here it was quite interesting to see that when we, choose to compare our data to the standard reference requirements that were, published by FAO regarding adults and older child, so child up to 12, 10, 8, 12 years old, it was found that this kind of new products are really, they are below the acceptance, for older child category. So, it means that the distribution of essential amino acids inside of this product was not sufficient for this segment of population. And this is quite an interesting point of view, take in account when you are formulating these new products. It's really, really important, as we said before, the not misleading message to give to the consumer, each consumer for different age. I'm going quite fast here because I know that the third The third speaker today will talk about part of these issues relating, the safety aspects to vegetal proteins. So vegetal proteins, we say many novel proteins, vegetal proteins have many kind of advantages using, but at the same time, there could be some compounds inside of vegetal proteins that can affect the digestibility. So, for example, the presence of anti-nutritional factors and like polyphenols and other protease inhibitors. This can be quite, quite, let's say, important to study, to trace, and also to identify in case, because they could be affect our digestion process. So, for example, nowadays, I think that the most common is soybean. Okay, we already know that soybean was maybe the first vegetal sources that was introduced in our diet. It's easy to handle because protein of soy are easy to handle to produce texturized ingredients and so on. They have a very, very high protein content, so quite comparable with meat one, and also for balanced essential amino acids and also nutritional value. But at the same time, soybean, as we know, can pose two risks for a safety point of view. One is the allergenicity. Most of the proteins in soybean are big allergens, so are known to be allergens, and also the presence of different proteases inhibitors inside of soybean. This can lead some problems, even if we know that by applying different thermal treatments, we are able also, most of the cases, to inhibit the anti-nutritional presence. But if we would like to compare the protein digestibility in a vegetal or alternative source to an animal one, what can we do? How can be the tools, the technical tools that we apply for this reason, and also to see how protein are accessible. Here are just summarized the impact of different modification of digestibility. So the structure of vegetal proteins, I've already told you. Proteins inside of a vegetal matrix are complex with, for example, the fiber structure of polyphenol, and this can make a protein less accessible for digestive enzymes. Not only vegetables, but also let's think about insects, which is a alternative animal alternative protein source. In this case, there is this polymer that is called chitin, and chitin actually is quite strong as an anti-nutritional factor. So the presence of chitin inside of insect powder, insect extract and matrices has been proved to affect the digestibility of protein. And also the other specific, I'm not going into detail because you will see some kind of more focused description in the next presentation. Technically speaking, and then with this part, I'm going to close my lecture. That was the need of try to mimic in vitro what is happening in our body, because you can understand that there are many in vivo studies, but it's quite difficult to follow in vivo the fate of protein inside. You can do in animals, we just mentioned before, but still is not so, cannot be accessible for everyone, for every scientific laboratory and group. So these guys, I think some years ago, it was just a European project. I think it was born as a cost action. And they released this InfoGest protocol. The idea is to to got an harmonized in vitro procedure, because many people have already started to mimic. I mean, if you think about it, you have just to identify which kind of enzymes are working during the digestive process. So in the first part in your mouth, so the salivary amylases, for example, what's happening in the gastric phase, you are decreasing pH, and pepsin, for example, is the most one you present. And then in the intestinal phase, pancreatine and trypsin and other enzymes. So if you know a little bit enzymatically speaking, let's say, how it's going on, you can think about reproducing in the lab each step. Okay, so that was the idea at the beginning, but each laboratory was doing by itself, and so then it could become very difficult if you want to compare different results, if you are compare, in our case, different protein extracts coming from different sources, if the technology that you applied to mimic the digestion process is not the same. So this InfoJet nowadays, I think it has become an harmonized in vitro procedure. Here is described the static one, but there are some improvements that have been made also to try to mimic the fact that we are the digestion in our digestive apparatus is not static. So there are also some examples of semi-dynamic or dynamic one. Here is, I mean, there is the reference if you would like to have a look. Here is the procedure. So you see, it's like a receipt. For us, it's quite a chemical reaction receipt, because you are mixing all the enzyme for each part and mixing together with the salts. Very important is to strictly monitor the pH, because you know that, I mean, pH is changing for each case. And then at the end, when you have finishing, you got your sample, and then you can analyze your results, and you can try to, I mean, to have an indication of the digestibility. So how the protein inside of your foods have been degraded, have been digested, fragmented, solubilized, and so giving rise to the production of amino acids and so on. Of course, also in this case, you have a picture, you got a picture of what is happening by here, so at your intestinal space, but you do not have, and so you will see which protein fraction has been digested and can be potentially be absorbed in the next step that we will see afterwards by our colleague Dimitris. But still can give you some information, I would say, also for a food technology point of view, to have an idea. Again, we were speaking about the possibility of mixing, producing different foods with different alternative and vegetable protein ingredients in order to increase the, for example, essential amino acid profile, but at the same time, by applying this protocol, we can have also an idea if the combination is the right one, and potentially we got a solubilized protein fragment, very high amount of amino acids, because the amino acids are the ones that finally, the last piece that is degraded from a protein goes into our body. Here is just a picture. I'm not going in detail, but I was just I just wanted to show you how, I mean, how can big, I mean, the part after, I mean, you got, I mean, we can mimic the digestibility, we can mimic in a flask in the laboratory, we can have finally our digestate, and then we can apply different techniques in order to better characterize the protein solubilized, and then see how many, for example, here is an SBS page. Of course, if you run an SBS page before and after applying your InfoGest protocol, hopefully you will see some differences. So it means that most of the protein should be disappear from the SDS page because they are digested, and this phenomenon should be increased starting from your foods arriving to the intestinal one. But then if you see some bounds that are still evident, like in this case, then you can give some hypothesis of the resistance of this protein. So this also could be very interesting to be studied. Also, the analysis of peptides and that are released is also can be has been done. Also, it is commonly done, and also in the past, because that could be also some indication what is the sequence of the short peptide that has been generated, can be potentially absorbed by your body. And if the sequence is the right one, we know that some amino acid sequence potentially are responsible for different bioactivities. So that also can be also an information that can be expanded for the next steps. For the InfoGest,
Tullia,
yeah?
we have to wrap it up now, unfortunately, because otherwise we won't have time for questions or break afterwards. But I just want to give you the last words if you want to sum up something. Thank you.
Yeah, I'll just leave you this slide is just an information about comparing the vegetal to the animal proteins by this technology. So just to conclude, I think that the option of introducing vegetal and alternative proteins in our diet is a very, very good option, and we have, of course, to explore it. I think still a little bit, mainly I would say for safety aspects, but considering the nutritional point of view, we have many tools that can be, I think, rigorously applied in order to get the final goal, which is to have a product that can be good for the consumer and also for a quality point of view, but also a nutritional point of view. Thank you for your attention.
Thank you very much, Tullia. It was a very comprehensive overview about amino acids and digestibility. So unfortunately, yes, Özlem has a question.
Yeah, if we have time, because you were just saying only one short question, maybe.
Only one short question, yeah.
Yeah, thank you very much. It is known that maybe the most important one is the amino acid score when we think about the protein quality, but it just goes in hand with digestibility, of course. Are there any systems where you think about alternative proteins that you have a really great amino acid profile, but the digestibility suffers? Are there any cases where you just say that, okay, there are some components, some definite proteins that just doesn't work really hand in hand?
But for example, the insects, substrate, you have a very high amount of essential amino acid inside. The distribution is really high. So if you compare it to soybean, which is it's better. But at the same time, it's quite a tricky technical part, the production of insect powder, due to the fact that the presence of chitin, for example, can alter, the digestibility, can affect the digestibility, and also can affect the total quality. So in that case, I would say that could be a problem, but still we have time to see.
Thank you very much for answering the question, and thank you, Özlem,
for
raising the question. If there are any further questions, you can write Tullia also an email or reach out to her directly. If there was a specific topic that interested you the most. So therefore, Tullia's contact will be in the chat. And before we go on with our next speaker, I would do like 5 minutes of a short break as a biological break. So in 5 minutes, so 10 minutes to 3 in German time, please be back when we hear from Dr. Dimitris Fatouros about bioavailability. And therefore, I kindly ask you to be back in 5 minutes. So see you then.
Is it okay now, Simon?
It's still loading. Yes, now it's in presenting mode.
So
Perfect.
now,
Thank you.
okay, when I move
Yeah.
from one slide to the other, it works.
Yeah, slides are also changing.
Okay, thank you very much.
All right. So welcome back, everyone. So in the next half of an hour, Dr. Dimitris Fatouros will talk about unlocking the nutritional potential, and I give the word to Dimitrios. The stage is yours.
Well, hello everybody. Thank you very much for the invitation. Actually, the title of my presentation is Unlocking Nutrients Potential, and we're going to discuss a little bit, or to describe, if you want, in vitro and ex vivo models, which mimic the GI tract for the absorption of biological active substances. So, a brief outline of the presentation is some general considerations of the GI tract anatomy, and also we are going to describe some in vitro and ex vivo permeability models, and then we will present some examples. I will start my presentation from the last slides from our colleague Tullia. So before moving, of course, to this kind of models, let's remember a little bit about the physiology of the GI tract. So actually it consists of enterocytes mainly, which dominate in the intestinal epithelium. And as you can see to the left part of the slide, these are columnar type cells, and they are held together by tight junctions. Actually, these tight junctions are regulating the diffusion of small compounds, and at the same time they exclude all toxic large molecules. You can see also here within the magnification we have the presence of microvilli, and these are between 3 up to 7,000, and these are like small hairs. They are on the apical part of the membranes, and actually they can benefit the absorption mechanism. Looking at the lower part of our graph to the left, you can see that we have here we can see here very nicely the enterocytes, and in between, of course, we have these tight junctions. So there are several ways that an active compound, either a small molecule or a large molecule, can pass through these membranes, because actually these are membranes in a general term. So we have the passive transport, which is from the apical to the basolateral side, and we can have either transcellular passport, which is the actives follow the transcellular pathway, so they just pass through the cell membranes. And also we have the paracellular pathway, where especially large molecules can pass through the intestinal wall, the intestinal epithelium, through the tight junctions. Of course, we have the active transport, which is carrier-mediated or vesicle-mediated. It's what we know, the pinocytosis. So a few words how we can calculate actually the transport of the APIs. Actually, the cells also is a membrane. So if we go back to the physical chemistry classes, we remember that the transport of actives through membranes are described by the Fick's law. And what the Fick's law says actually is that the steady state flux, which is the rate of transfer per unit, which is expressed in grams per centimeter per hour, and also is equal with the concentration gradient and over the linear distance which the molecules travel. And of course, the diffusion coefficient. And this is the steady state flux. However, the most characteristic parameter for all kinds of membranes, is the apparent permeability coefficient, which describes the amount of drugs crossing the barrier per unit and unit area, and actually it can calculate it by this equation. We will focus mainly to the paracellular mechanism, because this is the main way that the large molecules can pass through the epithelium. So actually this happens through the water-filled intercellular spaces, the tight junctions. And what happens? We have the loosening of the tight junctions, so the active can pass through these small junctions. However, we should all keep in mind that when we are working with permeability studies, and in some cases also we use permeation enhancers, at the end of the experiment we should be sure that the integrity of the membrane is still there. So that means that the presence of any permeation enhancer shouldn't change in a reversible way the function of the membrane. And this is the reason that we use a device which is called a transepithelial resistance device. You can see it's quite simple. It consists from a power supply and of course an electrode. So you can see here it's a typical transwell cell with CaCO2 with a cell monolayer. You can see also here the electrode, the anode and the cathode. The anode is in the apical side, the cathode is in the basolateral side. So in essence we measure the resistance or the difference in the voltage. Actually, transepithelial resistance studies measurements are used in order to monitor the quality of epithelial cells which are cultured on filter supports. So after all these few general information about the GI tract, I will move to the kind of models we have so far for measuring the intestinal absorption. Actually, I'm going to focus mainly to the intestinal, to small intestine and large intestine, and not to stomach or to the oral cavity. So we have the 2D in vitro models to evaluate the intestinal absorption. That means that we have the artificial models. In that case, we have synthetic models based on lipidic membranes, and these models offer a great reproducibility and stability. So in simple words, we have a polymer substrate, and we add some lipids which resemble more or less the ultrastructure of the epithelium. However, with this kind of models, always we have passive diffusion from one direction. We have also the to the cell-based models. In that case, we use the transwell cells again, but we develop a cell monolayer. We have the apical and the basolateral compartment, and actually the main advantage of this kind of setups is that we have influx and efflux, and in simple words, we can measure our active both in the basolateral and the apical compartment. So in this table, actually, we summarize the advantages and the disadvantages between the synthetic and the cell-based cultures. For example, very, very briefly, you can see that the synthetic models are, they have a low cost, they are rapid to use, they're easy to manipulate. However, they are coming also with disadvantages. They can hardly mimic the in vivo anatomical and biochemical structure of the small intestine, and also they can have the ability to utilize human cells. However, their main disadvantages is that compared with the artificial membranes, they have longer times of preparation. We have risk of contamination, and of course, we have higher cost. If we'll describe, if we'll stick also to the artificial models, I will give only a representative example, and this is the Pampa. You can see there to the right part of our slide. We have more or less, it's like a transwell cell. So what is the main advantage of the PAB actually? That it is a high throughput technique, because we have so many wells, so we can have so many repetitions of the drug or the large molecule to measure its absorption. So with the magnification, you can see here that we have again the donor and the acceptor phase. We have the membrane this time. This is an artificial membrane. So again, in a similar pattern and mechanism like the synthetic models, we measure the transport from the apical to the basolateral part. And it's Pamba actually is especially advantageous in early drug discovery, and this is something, at least from my experience, that companies really like, that they have to have high throughput techniques, because that saves time, and of course in the later stages, you can reduce the number of the in vivo experiments. Again, the same setup also could be presented. Again, these wells, but instead of using the artificial membrane, we can use Cacotum cells. So in that case, we have again all the advantages with some disadvantages when we have a cell-based membrane. So at least the home message from this slide is that high throughput techniques are really fast and are really of great interest for the industries, because you can in really short times you can produce a lot of data. Again, I will move to the 2-day cell models, and because we are working with formulations that are administered orally, I think most of us were familiar with the Caco2 cell line. It was isolated by Fogg and his colleagues, and it is from human colon adenocarcinoma. Is the most widely used cell-based model, which can simulate the physical and biochemical barrier of the human intestine, and most importantly, it can be used both for pharmaceutical and nutraceutical in the pharmaceutical and nutraceutical field. Also, it's really important because it's a gold standard for the regulatory authorities for ADME and toxicological studies, because you can get information for the absorption and metabolism of substances across the intestinal epithelium, and also we can have a kind of in vitro and vivo correlation with the data we are going to obtain in humans. Equally important, and this is the reason that I mentioned this example, are the co-culture models. And one co-culture model which is quite popular at the moment is the combination of cacotune cells which are co-cultivated with other type of cells. And I have here in this cartoon the first choice of this kind of co-culture models is the HMT29 cells. And what is the reason for that? Actually, you can see it here in the transwell cell. The co-culture of these two cell lines can produce mucus, and we know that mucus, this thin layer in the GI tract, is really very, very important for the transport of any active compound. So compared with the cacotune cells alone, the co-culture also produces this mucus layer, and of course, in this case, we can get more information about the transport of the API, because we have also the extra factor of the mucus layer. The next model that has been used so far are organoids. You can see here nicely organoids that have been developed in the lab. Actually, organoids are in 3 dimensions, organs that are created in vitro, and they have a realistic microanatomy due to their in vivo-like self-organization. Although these 3D structures have a lot of similarities, in our case, with the human intestinal complexity, because of their morphology and architecture, poses many obstacles when we would like to measure the transport of different actives in the GI tract. And this is due to the fact that we have the closed lumen, which actually precludes any direct access to the apical surface. On the other hand, one of the main problems is that we don't have vascularized network in this organoids, and also these organoids are statics, so we don't have a dynamic system which actually is coming closer to the in vivo situation. The next model that has been already used in the literature is the human reconstructed small intestinal tissues. You can see here a very nice image. We're going to discuss it a little bit later. And actually, one of the pioneers in the field is this company Matek, which develops a lot of human reconstructed epithelia. For example, nasal, buccal, vaginal, skin. In our case, the epithelium of interest is the intestinal epithelium. Actually, they can work very, very nicely for drug absorption and metabolism studies. And the most important thing is that they can be used also for toxicity and inflammatory studies. So actually, the structure of the epithelium is in that way that, for example, if we would like to get information for IBD or Crohn's disease, actually we can induce changes to this epithelium, so that will be a diseased epithelium, so we can get much more information. Again, the same problem we have with this kind of epithelia, with this kind of structures is the problems we faced with the organoids. We have absence of inner vascularization, and also this is employed under static conditions, and we don't have this dynamic physiological fluidic stimuli. Then the last part, which are much more complex compared with the other systems, are the 3 dimensions fluid dynamic in vitro models. Actually, these kind of models are developed for a lot of organs, but now I got some nice images for 3 dynamic fluid dynamic systems for the GI tract. So you can see, for example, in picture A, it's a little bit simple, but it gives you the principle. We can see, of course, it's a fluid dynamic system, so we have the flow, and on the upper part we have the laminar flow, the lumen, and this is separated with the capillary flow, which is the systemic formulation. In the second one, also you can see that we have this channel. In most cases, these are fabricated with printing techniques. So we can see we have the porous membrane, which is working as a substrate. We have developed the CACO2 cell lines, and it's really nice actually to see when the system is static and when the system is dynamic. When it is static and with this very nice confocal image, the main difference actually, between the static and the dynamic, we can see it here. If you can see it, of course, it's a little bit small, the magnification, but trust me, take my word, that in the dynamic we have also the development of this brass border area where we have the development of the microvilli. And this is actually the difference, because this is a static one and this is a dynamic. Again, if we wanted to make it a little bit more complex, we can see here that we have actually a gut on a chip. That means that in this case, we can develop the whole organ in a chip, and you can see here in very nice details all the main components of the intestinal epithelium, starting from the enterocytes. Goblet cells also are present, and Paneth cells also are present. And also moving to this graph, we can see again very, very nicely 2 different colors, the apical and the serosal, and these 2 colors also can show the different flows, so that means that this is a dynamic system. This is also something that is developed from a company, and if we move to figure F, this is a heat map when we can see gene expression of different APIs or macromolecules. So actually, why I have this figure here, because we can see that with this 3D fluid dynamic in vitro models, we can get a lot of information. We can extract a lot of information for our studies. And the last one, of course, again, it's from a company. Again, it's called, if I recall well, MIVO. And you can see when you have the MIVO, the static and the dynamic, when you have only the CACO2 cell layers, and when also you have developed the tissue. So you can see really nicely that most anatomical features are developed when we have the full anatomy of our epithelium. Very, very briefly, it's just for your information. You can see the characteristics of these fluid dynamic models, gut on a chip, 3D scaffolds, and so on. And actually, I have underlined, I have put in a frame, at least to my opinion, some critical characteristics that can actually improve the transport, which in turn can give us more information about our formulation or our macromolecule, whatever we would like to test in the GI tract. Another also nice system that I was working with when I was in the Netherlands, and I guess most of you are familiar, is with oozing chambers. Actually, oozing chambers took their name from oozing. He was a Danish zoologist. They are quite simple. You can see here the cartoon and then how they really like. We have side by side cells, so we have the donor and the acceptor. We have here in most cases a biological membrane, and I'm going to show you in the following slides some experiments we did at that time. But the most important thing is that at the same time we have two electrodes in the donor and in the acceptor. And what is the reason for that, to have these electrodes? Because actually we want to monitor over the experiment the membrane integrity of our tissue. In the literature, we can run experiments up, if I recall well, to 240 minutes using, for example, small intestine. After this time, nobody can guarantee for the integrity of the tissue. So actually, with these electrodes is a very nice way, because if we see any changes to the resistance, that rings a bell that there is something wrong with the tissue, and most probably we should stop the experiment. In the setup we had here, you can see here we had the electrodes, that was the donor and the acceptor, and here is the device which produce the voltage. It's really important that during the experiments, and this is, we can see it here actually with this tubing, we flush constantly our tissue with carbogen. Is a mixture with 95% oxygen and 5% of carbon dioxide, because we want to keep the tissue viable. And actually that was a modified version. This is the version that we can purchase from a company. So a few things about the tissue preparation. You can see we get our tissue in cold buffer, and then you can see that was pink intestine. In these experiments we were working with everted sacs, so actually we were removing a layer, and then we could open the tissue, and we could mount it to this side-by-side cells. Another also approach, which is also acceptable, and we do it also in our lab, is the everted gut sac model. More or less it's the same in that, like in the previous one we are working with diffusion cells. In this one you can see you get the intestine, you put it in a beaker, you tight from a stand. Actually, what you do, you make a sack, and into the sack you add your formulation or your protein, your API, and so on. And actually, what you do, it is thermostated, and you measure the transport of your substance from the sack to the acceptor phase. So over time, you can get a profile about the transport of your active compound. Again, you can see it here also. Here, these small sacks. They are just in tubes because we have special setup in the lab where carbogen also is flushed into the vials. Also, in this case, an experiment like that have a duration up to 240 minutes, because after this time, nobody can guarantee for the integrity of the tissue. And now I would like also to show you something.
Dimitrios, I just wanted to point out that we are getting close on time.
Okay, then.
Just to wrap it up, if you summarize your final words.
How 1 or 2 minutes?
Yeah, 2 minutes should be fine.
Okay, 2 minutes, very quickly.
Thank
you. Actually, Tullia described very nicely the infogest. In our lab, we have developed the in vitro lipolysis model, which is more for lipid-based formulations. So more or less, it has got the same principles. It consists of a double wall vessel, and you can see here we add our formulations. You can see we use also bile extracts of pancreatic lipase and also calcium chloride. It's kind of a reactor. So at different time points, we do sampling at 37 degrees, and we can measure either fatty acids or the API. I'm sorry, I will go very quickly. This is also oral delivery of macromolecules. This is coming closer to what we have done in our project. These are, you can see the intestinal bags. We used large molecules, fitzdextran, and also BCA. It's not in this graph, and we measure the transport across the membrane. We did also some studies with DTANI infusion and their components, and we did again permeability studies, and these studies are with rabbit tissue ex vivo. Also, I would like 30 seconds to say a few words for that, because this is something we have developed now in the lab. We are trying to coming closer to the in vivo situation. So actually we developed a chamber, and we have controlled humidity and temperature. You can see here we have a colon tissue, and we have also a holder here that was 3D printed, and actually we can do in real-time experiments, and we can receive the amount of the API that it is permeated across the membrane. You can see here the thermal camera. Very quickly, the last one is with nanoplastics. It's not so relevant, but I would like to show also that here we can get nice information with the ex vivo models regarding the molecular weight. So large molecules have very low permeation compared to others with lower molecular weight. And also in the lab, we do a lot of cytotoxicity studies, live dead assays, which is, I don't have the time to explain, apart from MTT assay, also ROS production and LDH. And the last one, because I saw some organ on a chips, we have developed also organ on a chip for transport of substances in the buccal mucosa, and now we're working in the GI tract. Very quickly, summarizing, cell studies can offer admin tox information, and artificial membranes have the ability of high throughput studies. In vitro digestion models, we can rank different formulations, and ex vivo studies resemble closer in the in vivo situations. Thank you very much, and I would like to apologize for if I'm out of the time.
No worries. Thank you very much, Dimitrios, for this very nice overview about hands-on quality evaluation on food. Therefore, thank you very much. I would still open the floor for at least one quick question, if there is any from the auditorium. Yes, Özlem, please.
I have many questions. Thank you very much, Dimitris. No, it was. I mean, it was really interesting to see that it's so much fluid dynamics is involved actually in that was a realization of really those systems. And the one that you print, like 3D printed system that you were just explaining, so you just developed it to mimic the flow dynamics in the intestine, right?
Yes, actually, yes. And we control also the humidity and the temperature over time. And at the same time, yes, we're trying now to do permeability studies. That was for muco adhesion studies, but now we move one step further because we want also to see the transport through colon. That was for colonic delivery.
Yeah, that's really interesting and also amazing to see how you can even see just over time how the concentration or hypermeability is changing, right? Okay. Yeah. Did you already have studies done?
No, now actually we published this study that was with 3D printing and the device, and now we are moving because we wanted to measure the active compounds. This is the next time.
Okay, great.
I can share if you want later the presentation with Simon.
Yeah, great. Thank you very much. Thanks a lot. I will check the study. Yeah.
Thank you very much, Dimitrios, for your effort. All right, then we come to our last presentation of this webinar, and I open the floor for Frederick Lia from Malta College of Arts, Science and Technology. Are you there, Frederick?
Yes, I'm here, Simon. Can you hear me?
Yes, loud and clear.
I will share my presentation soon.
Yes, thank you. And the stage is yours.
Can you see my presentation, Simon?
Yes, in presenter mode. That's perfect. Perfectly fine.
Thank you, Simon. So my name is Frederick Lia. I currently teach at MCAST in the Institute of Applied Sciences, and today we are going to discuss the toxicological aspect of the alternative proteins that we are proposing. Well, when it comes to the toxicological aspect, okay, one needs to take in consideration what types of toxicity are we talking about. So toxicity in general can be divided into two. We have the chronic and we have the acute. So acute toxicity is something that happens in hand, while chronic toxicity is something that actually takes time to develop. Now, let's go to the contents of what I'm going to present in this presentation. I'm going to give you a basic idea of what types of alternative proteins there are currently on the market. There are alternative hazards when it comes to these proteins, especially when it comes to microbiological hazards, including mycotoxins. We also go into a little bit more detail when it comes to the anti-nutritional factors, which are intrinsic factors which are found within the plant itself. We're going to discuss how we can qualitatively assess the presence of these anti-nutritional factors, followed by the quantitative aspect. And we're going to go a little bit in detail when it comes to the actual heavy metal toxicity, their health risk assessment, and also their safety considerations. Now, with respect to alternative proteins, there are currently 4 major pillars of alternative proteins that are currently being marketed. That is the plant-based proteins, the ones that we are currently working on, lab-grown meats, which they have their own toxicity on their own, considering that they are grown inside the laboratory. We also have mycoproteins and insect proteins, which are also amazing, which are also emerging in the current state. Now, when it comes to alternative proteins, it's safe to say that, yes, under controlled conditions of growth and rearing and harvesting conditions, followed with good preservation methods and good cooking methods and good sample pretreatments, they are essentially very safe to use, and they don't offer any form of human form of hazard. Yet again, it's good for us to understand how such potential risks may arise. So what are the potential risks that we can discuss? There is also, for example, allergenic potential. It's discussed by Tullia in this presentation, in her presentation. For example, we have cross-reactivity of allergenic behavior when it comes to chitin, which comes from the insect's exoskeleton. We can also have the potential of microbial contamination coming from either the primary source material or as during the processing itself due to lack of hygiene. And then we also have chemical contaminations, which include mycotoxins, which are derived secondary metabolites from fungi species. There are also heavy metals that might either originate from the plant itself, or they might have been introduced during the process of extraction. And then we have also the possibility of having plasticizers and on the other organic pollutants, especially when we look at insects, since the majority of some of the insects which are being proposed in the market are also able to eat up plastic. So they might also bioaccumulate plasticizers inside their body. With respect to the anti-nutritional factors can be broadly divided into two. The anti-nutritional factors that are heat liable and those that are not. So in the table, I have discussed the four major classes of anti-nutritional factors. And when it comes to heat liable ones, we have, for example, cyanogens, which are glycosides, which in the presence of enzymes of glucose, they release hydrogen cyanide. Those are easily removed once the product has been boiled or it has been left to steep. On the other hand, if we look, for example, for tannins on the other hand are very difficult to remove during heating. And in fact, heating also improves the release of tannins and other secondary metabolites in some instances. So again, based on the anti-nutritional factors and the standard safety assessments which are issued by the FDA, we have no issues when it comes to the actual potential toxicity of these alternative proteins. Now, when it comes to microbiological hazards, we assume that in general, we will operate under standard HACCP conditions. So we have special contaminants of bacteria that one needs to take the lookout of, for example, including Salmonella, Escherichia coli, and Listeria, especially when we're dealing with insects and plants and the presence of water and contaminated soil that can bring it out through the entire process. There are also spore-forming bacteria that one needs to take a good look at that include Clostridium and Bacillus. There are also, when it comes to insects, proteins, there are certain bacteria which are parasitic to the insect guts, okay? And one also needs to take them in consideration, okay? And also toxins, especially when it comes to lab-grown meats, there's also the potential of also having growth factors which can ultimately contaminate the end product. Remember that lab-grown meat are grown under tissue culture conditions, and we need to supply them with growth factors for the actual muscle to grow. Now, when it comes to mycotoxins usually are produced by three genera of fungi, okay? And they are mainly belonging to Aspergillus, Penicillium, and Fusarium. They can also have a potential of occurring in plant-based proteins due to the possibility of having a moldy product, whether now the mycotoxin is coming from the primary source material or whether it is the storage condition. One needs to always keep an eye out for these type of toxins. And especially when it comes to mycotoxins, they have been shown that they are more prone to grow onto other fungal substrates. So for example, if we have proteins which have been isolated from Saccharomyces, the standard yeast, the proteinaceous material over there also needs to be kept eye out for mycotoxins, as it might also proliferate their growth. Now, I need to remind you that the higher the protein content that you have in your product, the lower it needs to be your water activity. And it is there for a reason. The drier your compound it is, the drier the product that you're offering, the better it is, because it will not allow the growth of these particular fungi. Now, when it comes to anti-nutritional factors, which I will base the rest of my presentation on, no one can go without discussing the anti-nutritional factors before discussing the plant secondary metabolites. So what are we doing in this project? In this project, what we're doing is that we are trying to extract the proteins from the plant. And we know that proteins, carbohydrates, and lipids form part of the primary metabolism of the plant, which is the primary metabolism is the metabolism which allows the plant to survive. Well, the plant is a sessile structure, okay? So it needs some form of defense from herbivores and from other creatures. So it did develop a secondary type of metabolism known as the secondary metabolism. And in the secondary metabolism, whilst it's not essential for the growth of the plant, it does offer an evolutionary advantage. And there are a large number of different classes of plant secondary metabolites, starting with terpenes, which can be divided into monoterpenes, sesquiterpenes, and diterpenes. These are the volatile oils, which are characteristics of any aromatic plant. Now, moving away from the volatiles, if we look at the non-volatile types of metabolites that the plant produce, we have the phenolic compounds, which is the largest one of these classes. And these can include phenolic acids, flavonoids, stilbenes, coumarins, lignans, and tannins. Tannins, on the other hand, are further divided into two, the hydrolyzable and the condensed tannins, while on the other hand, the flavonoids are further divided into another 6 different classes, the flavones, the flavonones, catechins, flavones, anthocyanins, and isoflavonoids. And if we move away from the phenolics, then we also have nitrogenous-based compounds. And major class that we look for are the alkaloids, okay, such as caffeine and nicotine are nitrogen-based compounds which are found in plants. And under nitrogenous-based compounds, we also have the cyanogenic glycosides and the non-protein amino acids, okay? And then we also have another class of compounds, which I will not be discussing in this presentation, which are those belonging to sulfur-containing compounds, which are mainly found in the Crustaceae family and in the Brassica as well. The plant metabolites, which I would like to discuss a little bit more in detail, are tannins and saponins, and I would like to also discuss the presence of other anti-nutritional factors apart from the secondary metabolites. So secondary metabolism, are not the entire class of secondary plant metabolites, are not all of them anti-nutritional factors. It is purely dependent on the type of metabolism that you have and on the concentration you can have them. In fact, most of the phenolic acids under very low concentrations, they're actually beneficial to the organism itself because they act as antioxidants. On the other hand, we have another class of anti-nutritional factors, which are the organic acids. And these are the phytic acid and the oxalic acid, which we'll discuss a little bit more in detail. Now, if we are going to discuss the anti-nutritional factors which are present within the plant itself, one needs to take it also in consideration within the co-evolutionary context. So we are now taking out the primary source of energy from the plant. So the plant has to defend itself. So this is why it does produce some form of compounds which prevent it from being eaten. Now, in turn, okay, us who eat the plant have also evolved some form of enzymes which will allow us to degrade these compounds. This is the co-evolutionary behavior. So, for example, we cannot assume that a ruminant animal will have exactly the same effects as a monogastric animal. So our digestive system cannot be compared to that of a ruminant, for example. So a cow can eat much higher doses of these anti-nutritional factors without being affected. However, us, being a monogastric species, the presence of these anti-nutritional factors can greatly impart our digestibility. Now, again, under low concentrations, such compounds can be found actually to have a beneficial effect. It is only the high concentration which can become toxic. Now, let's start off with the most common ones. Let's start off with lectins. Lectins are nothing more than glycoproteins, and they are mainly found within 10 to 2% of the total protein content, which comes from food, for example, including beans. Remember that beans are seeds that inside them contain the embryo of the plant. So the plant itself needs to produce some form of substances which will prevent them from being eaten. Now, these lectins, on the other hand, are able to hemoagglutinate, meaning they are able to clump up red blood cells together. The presence of the glycoprotein side can also allow lectins to act as antigens to villi inside our intestinal system. So if in turn the villi also express a particular antigen, then we have also the problem of sudden allergenic behavior. Okay, so we have sudden cramps, and we also have, for example, bloating and gas if we, for example, have a high diet of lectins. Now, on the other hand, lectins are also able to arrive to the colon, where they can be, on the other hand, fermented by bacteria. If not, they can also cause an intestinal disruption of the mucosa, causing leakage of blood, but also secondary bacterial infections. Yet again, lectins being a protein, they can be easily be denatured, okay, by simply soaking and autoclaving the food products before extracting the protein itself. On the other hand, we have tannins. So tannins are complex phenolic compounds. They can be broadly described into two based on their monomeric structures. We have gallotannins, which we have gallic acid, which is combined with a sugar moiety. And we also have ellagic acid, which is also combined to another sugar moiety, and that forms gallotannins and ellagotannins. Now, in turn, polymeric structures of these will give us complex tannins, or else will give us condensed tannins. Now, tannins themselves, the presence of the large amount of hydroxyl groups present in there, are able to do two major things. They are able to combine with proteins. When I mean combine with proteins, these also include enzymes, which are present either in your stomach or within your small intestine. So they reduce also the effectiveness of proteases enzyme to act on the actual protein. Furthermore, the tannins themselves are also able to combine with certain minerals, for example, zinc and iron and copper. By complexing with these metal structures, they do not allow the absorbance of these metals inside the animal. On the other hand, we all know that tannins are very good they are good in low doses. Okay, they act as a very good antioxidant. However, one needs to keep in consideration that large amounts of these can also make the product be very bitter and astringent in taste. And this is the whole scope of having tannins for the plants, is that when you bite into it, you literally can stop eating the plant. That's why the most of the protein precipitation also occurs within the mouth when you're dealing with tannins. Now we also have saponins. Now saponins are made up of the molecule of saponins are made up of two structures. We have the aglycone, which is a steroidal part, which is the hydrophobic part, which is able to either be made up of steroids or a triterpenoid structure. And we also have a large conjugated sugar, which is bound to the aglycone, and that large conjugated sugar over there will also offer this molecule the hydrophilic activity as well. So we now have a molecule which shows an amphipathic behavior. Basically, it also is able to dissolve both in the aqueous phase and also in the non-aqueous phase. Now, this gives the saponin its actual name. Why? Because this activity, this amphipathic activity allows that when you shake a saponin, you will literally form a foam. And most of the saponins come from the tomentum, okay, which is the outside part of the seed, most of legumes and other seed oils, especially kidney beans, peas, and also chickpeas and broad beans. As you might expect, they taste horrible, because they have a very bitter taste. Okay, literally they taste of soap, and their soapy action also limits the digestion by other enzymes to the actual protein itself. So when supplements enter inside the small intestine, they can also inhibit the trypsin and the chymotrypsin, preventing the protein from being digested. Now, again, under very low conditions, they have also been found to lower cholesterol. So they are also being implemented into the food industry that they have been shown to actually lower cholesterol. Now, and apart from that, one can also functionalize the steroidal part to also employ them into, for example, advanced payload delivery for specific cells. Now, one of the more, let's say, less known part of the plant secondary metabolites are the presence of phytoestrogens. And phytoestrogens are compounds which structurally mimic estradiol, which is estrogen. So they are able to interact with estrogenic receptors. Once they interact with estrogenic receptors, they are able to form some form of hormonal changes. Okay. One class in particular which sticks out is the isoflavonoids, okay, which are derived from soybeans. While yes, they can have also a potential mitigating effect when it comes to cholesterol reduction and also alleviating the risk of menopause, one should also be considering the amount that one is eating. Now, with respect to the other anti-nutritional factors such as phytic acid and phytate, since we are dealing mainly with proteins which are extracted from seeds, we need to remember that the plant that produces the seed, okay, during the flowering, it takes large amount of phosphorus. And that large amount of phosphorus is in turn given to the embryo of the plant, which is the seed, to store it there so it can germinate. Well, the problem is that the plant does not store phosphorus as phosphate, but it stores it as phytic acid. And it's nothing more than a glucose molecule, which all hydroxyl groups literally have been esterified by phosphates. Now, this multidentate activity will allow, again, just like how we've seen in tannins, they will allow to complex specific metals like zinc, calcium, magnesium, and iron. So we start to see if we have a large diet of phytic acid, we start actually seeing the animal showing deficiencies in these specific minerals. Specifically in humans, zinc has been shown to actually have a great reduction when it comes to phytic acid. Well, the opposite happens when we're dealing with chickens. So for example, chickens in their diet, once they are supplemented with phytic acid, they actually grow bigger and they actually show less of an impairment compared to other mammals. So this is why we need to also consider the digestive system of what we're dealing with. Under low conditions, under low doses, for example, they have been shown to also reduce cholesterol and they also help to prevent against heart disease. And phytic acid is also the remedy can also prevent renal stones, which is caused by another acid, which we'll discuss further. Yet again, phytic acid can also be reduced by soaking, germinating, cooking, and autoclaving. So this acid itself can also be removed, around 20% can also be removed by simply autoclaving. Now, oxalic acids, okay, oxalates and oxalic acids, they are organic acids like phytic acids, but on the other hand, they are widely distributed inside the plant system. So oxalic acids in general, organic acids are used to translocate small molecules within the plant. Okay, and the plant produces two types of oxalates. Produces soluble oxalates, which include sodium and potassium oxalate, And it does also produce a large number of insoluble oxalates, which are those related to magnesium and calcium. And calcium oxalate has been found to cause kidney stones. Why? Because we are unable to metabolize this particular acid. It will start to bind to calcium inside the system, causing the formation of sharp calcium oxalate needles, which will cause pain during urination. Again, we do offer some form of quantitative test. So if we want to quantify the presence of these anti-nutritional factors, we need to decide whether we want to take a qualitative aspect or the quantitative aspect. So qualitative tests are very easy tests and are just purely give us the presence or absence. So for example, we have qualitative tests for phenolic acids, where we take the protein substance that is provided, we extract as much as possible, whether by boiling, pH adjustment, or doing a digestion. We remove the supernatant and then we test supernatant for the presence or absence of these particular compounds. So for example, if we want to test for phenolic compounds, we have an easy test using the ferric chloride test, where we just add a small amount of 5% ferric chloride, and what we see is a blue to dark coloration. On the other hand, if we want to assess for flavonoids, we replace the ferric chloride with 5% aluminum chloride, and what we see is a red complex, indicating the presence of a complex of aluminum with a flavonoid structure. With respect to tannins, there are a large number of qualitative tests that one can do, including the gelatin assay, but also there is also the 10% NaOH assay, which will allow the formation of an emulsion. In the case of saponins, these can be easily tested just by placing the plant material in water, shake it, and if you see a foam which stays for 10 minutes, it's a good indication that you have saponins. For alkaloids, we use the Drogendorf reagent, which is a potassium bismuthate iodide complex, and in the presence of alkaloid, it will allow the formation of a red-brown precipitate which falls at the bottom of the flask. So phytosterols, with respect to carotenoids and phytosterols, these are the organic fraction of the potential anti-nutritional factors. So the aqueous extract is further then extracted with chloroform, and the solution is filtered, and the product is the residue is evaporated. We add, for example, concentrated H2SO4 in the case of the phytosterols. We see a red color. It's a very good indication that is present. While in the case of carotenoids, we add a few drops of antimonide trichloride, and if carotenoids are present, they should give us a blue color change. Sorry, it will give us a blue, it will give us a change of color from blue to green, so from blue to red. Just like how we have qualitative tests, we also have quantitative tests. Yet again, when I'm saying quantitative tests, that I can express now the quantity of substance with respect to a known standard. Okay, so for example, we do have quantitative tests for example, total phenolic content. Yet again, these assays are prone to having, let's say, an overestimation of the large number of products that could be present. Why? Because these assays are not specific to phenolics themselves. So essentially, one can do a total phenolic content assay, and the results that he gets might not exactly match, for example, a profiling-based approach, which was done on an HPLC, simply because these tests have their own limitations. They are simple, but they have their own limitations when it comes to sensitivity itself. So for example, in the case of the phenolic content assay, it is well known that, for example, sugars and proteins might also interact and give us a false positive when it comes to the Follens reagent. With regards to tannins and oxalic acids, we do have titrimetric methods, but they are extremely lengthful. They're doable, but they are extremely lengthful. So for example, with respect to the tannins, it's a titration involving KMNO4 and using indigo. It's a back titration of KMNO4 with indigo carmine indicator. With respect to oxalic acids, we had to first boil off the plant, precipitate the oxalate with the addition of calcium chloride. We'll form an insoluble calcium oxalate, and then we back titrate the amount of oxalate that we get with, for example, KMNO4. Now, due to this large process of huge, extensive downstream processing when it comes to the analytical side of these tannins and oxalic acid, we are actually opting to go for an ELISA-based methods, which are far much more sensitive, okay? And nonetheless, we can also quantify specific secondary metabolites using an LCMS-MS. The rest of the presentation, I will discuss now heavy metal contamination. So what are heavy metals? Heavy metals are metals which belong to the transition metal series, which are can cause potential harmful effect to the human being and to the animal consuming these metals. There have been studies in 2024 which showed that, for example, insect-based products have been found to contain, for example, concentrations of lead and cadmium and nickel, which were above the specified European limits. Yet again, we do have some issues when it comes to the European limits, because we work with the European limits. However, the FDA limits are not the same, for example, as the European limits. So different countries might have different regulations and different limits for different metals. And yes, heavy metals can originate from two major sources. So whether they come from the processing itself, whether they come from the salt which was used to precipitate out the protein, from the milling of the protein itself, leaching of the material. But one also needs to consider that the plant itself is also able to bioaccumulate special metals. So especially when it comes to legumes have been shown to have high probability of bioaccumulating lead and aluminium, which can pose a risk to both adults and children. There are other environmental effects, such as the area where you're growing the crops, if it is near to a heavily traffic area, for example, whether there is where there is an oring company next to it. So these are the things one needs to take in consider when we're dealing with heavy metals. Now, how do we analyze heavy metals? There are various ways one can analyze heavy metals. The easiest one of all will be EDX-XRF, which stands for electron dispersive X-ray fluorescence, which I didn't discuss in here. Another one will be the use of atomic absorption spectroscopy, which I'm going to present. And derivatives of atomic absorption spectroscopy also include graphite fairness atomic absorption spectroscopy, which is better than AAS because it offers a better sensitivity. And nowadays, there is also the introduction of ICP-OESs, which is inductive coupled plasma MS, which also offers a very rapid analysis for heavy metals. So since we just have an AAS, what we've done with my team, we digested a specific amount of protein sample using a digestion medium containing hydrogen peroxide, HCl, and nitric acid. This would allow literally to digest the entire protein, liberating all the metals inside solution. Now, since we know that when it comes to the analysis of heavy metals, especially when it comes to biological substances with dealing with proteins, we do tend to have something known as a matrix effect. We opted to go for the method of standard addition. So what is the matrix effect? So if I do a calibration curve of my standards in just water, my gradient will be one number. If on the other hand, I will do a matrix match sample, my gradient will be actually lower. So the sensitivity, the matrix itself is lowering the sensitivity of the output of my reading. So one way to go about it is, yes, it's an extremely painful job to do because each sample needs to be analyzed at least 15 times, but we build a calibration curve onto the sample itself. And we then bear backwards and we extrapolate the concentration of heavy metals we obtain. For a realistic value of concentration, we then further check our calculations. We also do the calculation of the standard error to check the reality of how much, whether the value that we obtain it is significant or not, different from baseline or not. So for
Frederick,
the,
excuse me.
yes,
We
2 minutes,
are
Simon.
getting close on time.
2 minutes.
Yeah, perfect. If you could
2 minutes.
wrap it up. Thank you.
2 minutes. So with respect to other methods, we actually did this following exercise for the 13 alternative proteins which were provided, and we calculated lead, nickel, cadmium, cobalt, and zinc. And we have presented all these over here. We then went ahead and compared them to the European legislation, and most of them fall within the spec by the European legislation. And we then went ahead to also have a computational approach. What is the computational approach? Once we determine the entire concentration of these metals, we then put them into a computational algorithm, which will give us an indication of how hazardous they are at a specific rate of ingestion. So we calculated our rate of ingestions based on 30 grams of proteins a day. And what we've seen that neither the hazardous quotient nor the hazard index are higher than 1. In fact, I calculated that we need at least to eat 2 kilos of proteins every day for us to start having some form of toxicity from these special types of protein. So in terms of a general conclusion, when it comes to the toxicological aspect, one needs to take in consideration the entire process of manufacturing, but also keeps in consideration the plant itself and the material that is being used, because different plants produce different secondary metabolites, and this is one where one needs to keep in mind. So thank you. Sorry if I went over time, Simon.
Thank you very much, Frederick, for wrapping up all of our today's topic within your final presentation. So everything came together really nicely. And I just want to open up the floor if there is any question for Frederick from the auditorium.
I always have. So I start, let's see. Thank you very much, Frederick. It was really nicely structured, could follow really all the details. One question, it is now for the heavy metal that we have really those critical limits and so on. What about for the anti-nutritional factors that you were mentioning, like phytic acid and so on? Since it cannot be just really like a limit, but how do you just deal with it? Because then we have to just give some, I don't know, like safety limits for this. How do we deal with it in European Union?
No, I don't know. To be honest, Oslim, the limits on phytic acid, there aren't any specialized limits, not even on oxalic acids.
Yeah, but there are some recommendation values.
There are recommendations, yes, there are recommendations, but I don't know the limits on the top of my head right now.
Yeah, because it is changing so much. That's the thing. I mean, it's just like so complicated since of the natural materials.
Because then you need to describe between phytic acid, phytate and phytite. And then it gets too complex.
Yeah, okay. So then,
but as long as we are pre-treating the samples with water, if we are autoclaving the sample, I don't think we will have such a large amount of concentration in them.
Yeah. And
what I'm not sure about is the presence of oxalic acids and tannins. Though.
Yeah.
Because tannins are very difficult to remove unless you bleach.
Yeah, I mean, we have some of the technologies that we are using actually to extract proteins. There are some examples that we did with Megit as well.
And we also have a problem because especially if you are extracting under alkaline conditions, you might also enhancing the extraction of other secondary metabolites. So you increase, for example, let's take phenolic acids, because I've published a study lately on orange peel. So if I change the pH, I increase the pH up to 9, yes, true, I have a higher protein content. But on the other hand, I'm extracting more phenolic acids because they become soluble. When they change into the salt format, they become aqueous soluble.
Yeah.
On the other hand, the alkalides will not be affected because they will not be soluble.
Yeah, we will discuss this on Monday further.
No problem.
Yeah. Thank you.
Right.
Perfect. Thank you. Thank you very much. All right. Then coming to an end of this webinar. First of all, I want to thank all of the presenters taking the time, preparing the presentations, and giving such a great overview from over digestibility, bioavailability, and toxicology. And I'm certain that it's very important when you think about food, and especially thinking about product development, then that not only taste is king, but also you want to create something healthy that does not harm anyone. Therefore, this is more or less the standard for anyone working in food-related industries, to take into account what is about your food quality. And therefore, I want to thank Tullia Tedeschi, Dimitris Fatouros, and Frederick Lia taking their time and giving such a nice overview. Thank you very much, everyone.
Have a nice evening. Bye.
Thank you very much. And if you want to stay in contact with us, please follow us on the following social channels, and there will also be the outcome from this webinar where you can find it. So thank you very much, everyone, taking your time and participating at this webinar. And I wish you a nice evening. And
Bye.
thank you, and see you hopefully soon.
Thank you all as well for organizing, Nija and Simon. Thank you very much for the good organization.
Thank you very much. Very welcome.
Thank
Enjoy
you.
your evening, everyone.
Yeah.
And have a great time.
Thanks. Bye-bye.
Goodbye.
Bye.
Bye bye.